Welcome to The World, the Universe and Us, the weekly news podcast from New Scientist. I'm Dr. Rowan Hooper.
And I'm Dr. Penny Sarche. On today's show, the incredible story of the mice with two fathers. We're reporting on the breakthrough that has led to their birth and what it means for humans.
We also report on a new study showing how and why a power nap boosts your brain power. And we've got scientific tips on how to get to sleep. We're also going to hear about a lizard species that survived to the asteroid impact that killed off the dinosaurs.
Amazing.
I want to say it's miraculous that they survived, but we're a science show.
We don't believe in miracles.
No, we don't. Okay, we're going to start with the news that for the first time, healthy mammals have been born with two fathers and no mother. This picture I'm looking at now, it's of two normal healthy mice, but they have two dads and no mother.
So when you say no mother at all,
no genetic mother,
right.
It means. Yeah, so there's a foster mother, there's an embryo that's made and they're grown in a womb of a female, but then they have no genes from her at all. Made from two sperm fused together.
Amazing. Interesting.
Yeah, it's called androgenesis, mice with two fathers, and that's what we're getting into this week.
Yeah, and I guess the immediate place that your mind goes to is children produced without genetic contribution from women or females in mice. What does that mean? Like, what could it be used for?
Yeah, well, I mean, the place you go straight away is, the market for gay men. There's an increasing number of gay couples who have children with, you get an egg donor and surrogate mother. But in those cases, of course, the children are only genetically related to one of the dads.
So potentially might this breakthrough mean at some point male gay couples could have a child that has a mixture of both of their genes? So yes, this step does seem to bring that closer, but we've got Michael LePage here who reported on this for us to tell us all about it. Hi, Michael.
Hi, hello.
Yeah. So Michael, this was a team in China, wasn't it? Can you tell us what they did, how they created this mouse that has two fathers?
Yes, so what they did is they took mice eggs and they removed the DNA from them. That's the set of chromosomes that would normally come from the mother. And then next they took two sperms and added those to each empty egg. So that had two sets of chromosomes like a normal embryo, but because they both came from males, that embryo wouldn't normally develop. But what they then did is they reprogrammed the genes on those chromosomes so the egg could develop, and the end result were two mice that were apparently healthy.
So there's been lots of little stories in this kind of area over the last decade or so. This isn't actually the first time that a mouse has been born with two dads. Is it? What's the new thing here?
Yes, no, there have been a few cases before where people reported the birth of mice with two dads, but in all those cases, those mice have either died very young, or there was one report earlier this year where they did survive to adulthood, but they were infertile and they weren't very healthy. So obviously it wasn't a complete success. Now, in this case, these mice are fertile. They've had, reproduced normally. I think the other significant thing about this is they've used a method which is potentially applicable to humans. So that's the big new thing here.
Okay, so we'll get onto the method in a sec. But first, let's think about the problem. Is it with having two fathers? Because, it's a first in mammalian history that you have a descent. Everything until this point has been it's come from two a male and a female. For millions of years, and now we've changed that. So that's a big deal. But apart from that, what are the problems with having the abnormal gene activity going on through these by fusing two sperm?
It's all to do with this phenomenon called imprinting. So obviously we get one set of chromosomes from our mother, one from the father, and in theory those are just two sets of DNA that are exactly identical, and they are, in terms of the sequence, they could be identical, but actually it turns out that there are these extra labels that are added onto them. And so when the eggs are being formed, the mother will add labels that sort of say, use these genes, don't use these genes.
Depending on maybe if she's hungry and might be a famine on, or a heat wave or something like that.
That's epigenetics. That's epigenetics.
Well, this is epigenetics, but these marks are made in every mother regardless of the environmental circumstances.
Okay.
These are sort of more fundamental than that. And
Okay.
we'll come on to that later.
Okay.
We're not going to go into that now. And then obviously with sperm as well, you get these sort of labels being added to the DNA, but with the paternal chromosomes, there are a different set of labels are added to sort of different genes, so the effects are different. And you need those two sort of complementary sort of sets for the embryo to develop normally.
So how did this team then manage to overcome this problem with imprinting?
This is my favourite CRISPR.
CRISPR.
So in this case, you know CRISPR for gene editing,
Yes.
of course. But what you can do is you can take the CRISPR protein and modify it. So instead of changing the sequence of DNA, it changes these epigenetic labels. And so this team...
So it is epigenetics.
It is epigenetics, yes. And this team used this to change those sort of labels on the genes so that they were more like normal, so that you had the sort of two paternal chromosomes, but then they were modified so they look more as they should do if it was a maternal and paternal set.
So
Okay.
even though this is a gene editing tool, that you're not actually changing the DNA here, you're changing like the markers that sit on top of the DNA and determine whether genes are active or not.
Exactly. This is something that's called epigenome editing, and it's a really promising development in and of itself for treating diseases, but in this case we've used it
And also it means you're not mucking around with the gene line. So that's why people think it's more acceptable to do it.
Exactly. So with the cases I mentioned previously, the earlier attempts to create mice with two fathers, they instead of trying to actually correct the epigenetic labels, they just modified the genes to get the same effect. And in that case, obviously, if you did that in people, you'd have genetically modified people, and that's a no-no. But with epigenome editing, you're actually correcting the problem directly. You're just changing these labels. And so potentially it's something that could be used in people.
Yeah. Not quite yet, though.
No. So the big problem is, so in order to create these mice with two mothers, I'm just looking at the numbers here. So yes, they had to implant 259 embryos in the female mice to get the two healthy mice.
259 just to get two healthy ones.
Yes, and it gets even worse. And to generate those 259 embryos they implanted, they had to start off with 587 eggs.
Wow.
Now, anyone who's familiar with IVF will know that, during a typical IVF cycle, a woman might get 10 eggs if things go well. So if you've got a process that requires hundreds of eggs, it's a non-starter for people.
It's like when we used to talk about cloning a lot, didn't we? Like 10 years ago or something, when that was all, after Dolly. And that was an incredibly wasteful process. I can't remember how many embryos it took to get Dolly, but a lot, probably similar number.
It was in the hundreds, I
Yeah.
think. Yeah.
And it's not got a lot better cloning.
No.
We haven't really improved it much.
So why is the success rate so low?
Well, for starters, you're putting two sperm together.
Yeah, it's not a simple thing to do.
Well, but if you think about it, a sperm, two sperm they can both have Y chromosomes, and you've got a YY embryo which cannot develop normally. So that's a quarter of the embryos are gone already.
Right.
Now, in theory, you could use techniques called sperm sorting and stuff, so you could probably improve on that. Then the other thing, this epigenome editing technique that I was talking about, they actually looked at how well that worked, and they were able to say in a lot of cases they were trying to change 10 sites in the genome, correct the labels at 10 sites. In a lot of cases it didn't work at all of those sites. So you'd need to get the efficiency of that up to improve the success rate. So, I mean, in theory that could be done, but it's not going to be easy.
So we mentioned that this isn't the first time we've had a mouse born from parents of only one sex. And for anyone thinking, but what about two mums? Actually, back in 2004, there was a mouse named Kaguya born, and that had two mothers.
Yeah, it's a nice little story as a digression here about Kaguya. It was named
I knew you'd mention this. I knew you would.
Well, of course. It's from a famous story in Japanese folklore called The Tale of the Bamboo Cutter. And in that story, the bamboo cutter finds a baby inside the bamboo stalk when he's cutting it.
Yeah.
And she turns out to be Princess Kaguya, and she's from the moon. And it goes on to have lots of adventures. But in the case of the mouse called Kaguya, that was a fertile female mouse.
Yes.
So we had then a female mouse, a fertile mouse with two mothers, and that was 2004. 21 years it's taken us, and we've only just got to the same stage to get one with two fathers.
Yeah, so it turns out it's a lot easier to create mice with two mothers than two fathers, and that's because you have to correct fewer of these imprinted genes in order to succeed. So it's a numbers game, basically. And in fact, this technique of using epigenome editing to correct the label, so this same team in China that created the mice we've been talking about, in 2022, they used this technique to create mice I sort of want to say with two mothers, but there's a twist to this. They actually created mice that had just one mother and no father.
Oh gosh.
So, yes, it's a
clone.
It's not technically. It's kind of a clone, not technically a clone.
This is parthenogenesis, right? So some animals do this anyway.
I see. So it's shuffled.
Yeah, you sort of you're not identical to your mum. The genes have been sort of shuffled around a bit, but you only have one genetic parent.
That's exactly right. So when an egg forms, there's normally an extra set of chromosomes that's spare and gets chucked out, and they put that back in to create these mice. But obviously that technique would potentially be relevant to lesbian couples who would like to have children that are genetically their own, and in theory that should be easier to achieve than with fathers, but there's still huge sort of issues with the safety and the success
And they could
rate.
only ever have girls.
Yes.
Yeah, unless you really monkeyed around with the chromosomes. Then.
Invent a Y chromosome.
Yeah.
Mm-hmm.
Genomic imprinting, I think, is one of those really interesting little quirks of evolution. Am I right in thinking that the reason the male chromosomes and the female chromosomes have these different sort of epigenetic markers on, it's a way for them to sort of fight things out and fight over what the embryo gets?
Well, we don't know for sure, but this is a theory. So it's like the father is sort of boosting genes that make the embryo grow faster at the expense of the mother, and so then the mothers fight back by toning down those genes, and so you have this more and more genes getting imprinted, and the end result is no change at all. But you've got this strange phenomenon going on,
running forwards to stand still.
But like the placenta is a real battleground for that, because most of the genes that drive the growth of the placenta are male imprinted.
Yes, yeah.
Because the male wants to get more nutrients into the developing embryo than the mother wants to give.
But if a mother gives too many, she might only ever have one child instead of
so
3 or 4.
It's an amazing conflict, isn't it?
Fascinating. Wake up, Rowan. It's time to discuss sleep, hard in the sleepy glare of the studio lights.
I can sleep anywhere, actually.
Can you?
Yeah.
Now that is a skill.
Yeah, I do like.
Skill for life. And sleep is, of course, one of the single most important things that you can do for your health and well-being.
And for your cognitive powers, and that's what we want to get into today.
Yeah.
You know how people always say, go to sleep on a problem. If you're struggling with something, you make a decision. There's a new study out this week that investigated the best kind of nap you can have to boost your brain power and solve problems. And we're going to get into that.
Yeah, and we're also going to review some research on how to get to sleep quickly for those who have trouble dropping off to have that power nap.
Yeah. So as I already alluded to, as a middle-aged man, I do love a power nap. Do you like a power nap?
I'm so sad at the lack of naps in my life.
Michael, do you like power naps?
I'm not in the habit anymore, so I find if I have one, I just feel very disoriented.
So you're not doing it right. You're not doing it right. And this is what today's about, right?
Yeah, okay.
So I set a 22-minute timer
22
for a power
minutes.
nap. Yeah.
Okay.
Yeah. So 2 minutes are to get to sleep. And I told you I can go to sleep well.
That is, I can never get to sleep in 2 minutes.
And then 20 minutes of sleep. And that's important.
And that's specific.
Yeah.
Why 20 minutes?
Well, so research has shown that it gets you into the right level of sleep. I always thought it was just the right amount of sleep, but it's the right level of sleep that you get to without going too deep into sleep that you wake up really groggy. So if you had an hour's sleep, you'd be in deep sleep by then. And then it wouldn't be, you'd be like, oh. But if you have a 20-minute nap, you get into the right level, that works. And also it fits into a, lunchtime.
Handy.
if you need to.
Are you sneaking off having power naps?
Sometimes, yeah. It's in lunchtime, it works.
Right, so it sounds like you've put a lot of thought into optimizing the power nap.
Yeah.
What's new?
Well, so what's new is the research this week that shows the exact level that you need to do. So I'd sort of blundered along to it, but this research shows that you need to get to the N2 phase of sleep, which is the first of the deeper phases of sleep.
So remind me again, because there's so many different phases and types of sleep. What's N2?
So you've got waking, then you've got the hypnagogic stage, which is very quick. We're going to come back to that. And then the first proper sleep stage is N1. That's just light sleep. Then you go into N2, which is deeper sleep, and N3, which is the deepest sleep. And all of the N's are for because that's it's called non-REM sleep.
Yeah.
And then after N3, you get REM sleep, the rapid eye movement sleep.
That's like the dreams.
Yeah.
Yeah.
And then after the whole cycle starts again
Yeah.
in the night, and you do about 3 cycles of that in every night.
And do the cycles get longer or shorter as the night wears
they
on?
change, and in the end you get more REM sleep in the morning.
Right, okay, yeah. So this new research shows then that what you want to get to is that N2 sleep.
So yeah, exactly. You want to get to N2, and the new study this week is that researchers happened in Hamburg. Researchers recruited people into a sleep study, had them follow instructions for a test about tracking dots on a screen and responding to the dots on a keyboard. But they left out a trick that made the test easier.
Ah, okay. So what they did was they had these volunteers do the tests with these dots 4 times. Then they gave them a 20-minute nap, not a 22-minute nap, sadly. And they were hooked up to an electroencephalogram while they slept. So that's one of these caps that measures brainwave activity. And then this is where the magic happened. After the nap, the subjects were tested again. And just over 70% of the participants had an aha moment. They figured out this unmentioned trick, and it made the task easier for them.
Yeah, so they were more likely to have this breakthrough in the trick if they'd gotten to N2, and they were able to confirm this with the EEG. So if you made it into that first period of deeper sleep, that's where the sort of extra ability came from.
So like the nuances of this, everyone improved at the task after a bit of rest, so rest is always useful. But of those who stayed awake, only about half figured out this trick. Of those that only got to the light N1 sleep, that was more like nearly 2/3. But then those that got to the N2 level, it was more than 85%. So that's really interesting.
Yeah. So I didn't expect that there would be that difference in cognitive benefit. Because like you say, I felt like it's just the rest that gives you the benefit.
Yeah. I mean, I guess it still is rest. Is it a deeper form of rest?
It's a deeper kind of rest. It's something's going on restoratively in the brain that helps you. And I always thought it was not that stage where you got this cognitive or the creative breakthrough. I thought that was the hypnagogic stage, which is that bit at the last border between wake and N1 sleep.
Yeah, and so this is like quite famous. A lot of people in history have tried to like emulate that phase, right? And it's where it's a bit hallucinatory and hypnagogic jerks occur, like where you feel like you're falling over and all of a sudden you wake up. Yeah.
So that hypnagogic jerk, what happens there is that the brain is sort of winding down. And the part of the brain that controls your movements is kind of going offline. But there seems to be sometimes this last spark of action in that brain, and that's what literally causes your muscle to twitch, and your leg might kick out or something, and then you incorporate that into your dream. So that the brain can make sense of it. So I often think, I've tripped up on the curb or something.
Yes, fallen downstairs.
With me, I've been flying and then suddenly I plummet.
That's a nice kind of dream. We have to talk about dream flying at some stage in the show. So you get this dream incorporation going on.
So very interesting. But what's the connection there between this hypnagogic state and creativity?
Yeah, no one quite knows why, but you do get this very stream of very rich imagery in it. But the problem is that because it only lasts maybe a few seconds, you normally carry on into sleep and then you forget all those images, or you might not have even been aware of them. But if you know they're there, you can start to notice them, and that's what I definitely encourage you to do. Like be aware of them. And it's only if something drags you out of this borderland, then you can remember them. And that's why, as you were saying, people have tried to kind of be in that state and wake themselves up before they go in it.
The famous one here is Thomas Edison he did something with holding balls.
Yeah. So he'd hold 3 metal balls in his hand, going to sleep, and then when he would just finally not go into N1, the muscles relax and the balls would fall on the floor and clatter, wake him up, and he would remember the hypnagogic flow.
And does he claim to have had any moments of inspiration from that?
Yeah, well, I mean, he did pretty well in his life, didn't he?
Shades of Inception there.
Yeah.
So you know a lot about this. Have you tried to fall asleep holding a handful of metal balls?
I haven't done it like that, but I did go to a sleep lab, and they tried to induce the hypnagogic and lucid state, and that works. And you can get to it through meditation, actually, which is another really, really cool thing to do. But like I say, once you know that it's there, you can kind of see it going on as you go into sleep. And it's a really interesting
overlap with like lucid dreaming as well.
Yeah.
Okay, so the takeaway from this bit of research then is that getting through the hypnagogic state, through N1 to N2 sleep, really helps you to have those aha moments of inspiration that can help you solve a problem that you're sleeping on. But what if you struggled to get to sleep in the first place? And so this week we've had Helen Thompson writing for us about what sleep scientists recommend if you have difficulty sort of drifting off.
Yeah, and is that there's a lot of sleep hygiene stuff they call, isn't there? Like blackout blinds and no caffeine.
No phones before bed and all of that kind of thing. Yes, that's all good advice, but there's also a cognitive technique you can try. It was developed by Luc Baudouin at Simon Fraser University in British Columbia, Canada. And the aim of this is it's intended to help you quieten your mind and let you fall asleep by mimicking the hypnagogic state that we've been talking about.
Okay, go on, tell me.
So think of any word.
Microphone.
Oh, that's a long one. Okay.
Why did you think of that?
So what you do now is you go through the letters of the word microphone, but for each letter, you try to think of as many words as you can that start with it.
Oh, God.
Yeah, so go on. M.
Mother.
Yeah.
Mint.
Yeah. And so the idea is that as each of these words comes into your mind, maths,
yeah,
music.
I'm asleep.
Well, yeah, you really spend some time picturing it, the image or the concept. And the idea is that's a bit like the spontaneous images that sort of start free flowing and coming to you when you're in that hypnagogic state. And apparently people who sleep well often report that they have this kind of hallucination experience of all these images before they fall asleep.
Yeah.
Which I think I know that I've done that. But people who really have problems sleeping often find themselves apparently like making plans or like actively trying to solve problems as they're trying to fall asleep, which obviously is not great.
Yeah. That's really interesting because the hypnagogic state normally is subconscious. And that's the mad thing about it, is that it appears, you can almost see it like a river flowing past you, all these images, and going, where are they coming from? They're just coming.
Yeah.
But this seems like a way of hacking it and forcing it to happen
by
starting off with images and thinking of them. I wonder if once you've started it, it might just take off on its own.
Yeah, I'm definitely going to give it a try the next time I need to. It should be said there isn't like big gold standard evidence research kind of showing that any of these sort of cognitive techniques really, really, really work. But there was a small study that found that students, for example, who'd been struggling to wind down to sleep, did seem to get to sleep more quickly when they tried this particular technique. We have a story now about one of the biggest disasters ever to befall our planet and how something actually survived. 66 million years ago, a giant asteroid hit our planet in the vicinity of the Yucatan Peninsula in Mexico. It left a crater over 150 kilometres wide and caused the extinction of most of the animal and plant species that were living across the world at that time. There's a village near the crater called Chicxulub, and that is what the asteroid is now referred to, the Chicxulub impact event.
I'm just going to call it the dinosaur-killer and not the Chicxulub event. But yeah, that impact, it brought down the curtain on the dinosaurs, as we all know. They'd had a good run, 200 million years, roaming the planet.
I can't help it, Rowan. The dinosaurs didn't go extinct. Birds.
Oh, we'll get to that.
Birds are dinosaurs.
Yeah,
They're here.
The non-avian dinosaurs
Thank you.
had their day. They were wiped out. And it ended their whole geological period, the Cretaceous, all over.
Yeah. Although, there were also climate change and volcanoes going on at the time. But clearly this asteroid did have a big, massive impact.
As it were.
Yeah. Impact in both senses of the word. And the news this week, though, is that a species of lizard that is alive today, its sort of ancestry or family, turned out to have been living in the actual vicinity of this asteroid impact and somehow survived.
Yeah.
And that makes them the only terrestrial vertebrates in the region of this impact that somehow lived.
So indestructible lizards.
Yeah, it seems to be. So these are called xanthusid night lizards. It's a misnomer, they're not actually nocturnal. And apparently you can find them in Cuba, Central America, and the southwestern United States. Now, what's happened here is a group at Yale have used DNA sequence data for xanthusids to make an evolutionary tree for these lizards. And they found that they could trace their most recent common ancestor of all the ones that are alive today right back to deep within the Cretaceous period, over 93 million years ago.
Wow. So basically they were around and thriving way, way before the asteroid came along.
Yeah. And, if you think about reptiles, these are lizards, they probably only had clutches of 1 or 2 offspring. They were slow to reproduce, and that's important.
Yeah, well, so how did they survive?
So many night lizards live in rock crevices, and their slow metabolisms are comparable to those of other survivors of other mass extinctions, like turtles and crocodiles. So perhaps they were able to sort of shelter during the impact and its terrible aftermath and just kind of live very slow while they hid out.
Well, the birds took off.
Yeah, and the mammals,
So what's interesting is we kind of talk about this asteroid so much, and I feel like it's lost its impact a bit. In terms of we just take it for granted, really.
Yeah, like you learn it in primary school now, right? Yeah. The asteroid that killed the dinosaurs.
Yeah. But I spoke to Steve Brusate about this. He's professor of paleontology at the University of Edinburgh, author of loads of great books. The Rise and Fall of the Dinosaurs is the first one I read of his. And when I spoke to him, he really put the survival of these lizards into context. So here he is.
I think it's incredible to envision these lizards surviving the asteroid impact that killed the dinosaurs 66 million years ago. Now, it is a little bit of an extrapolation. Of course, we weren't around to see these lizards survive. This is based on where the lizards live today and on their family tree and predicting that they were in the area around the asteroid when it hit the Gulf of Mexico 66 million years ago. But I think it's a very provocative finding, very interesting, because it makes us reconsider what it takes to survive an extinction. This asteroid impact was so brutal. This was a 6-mile wide rock that fell from the sky, traveling like 10 times faster than a speeding bullet, smashed into the Earth with the force of over 1 billion nuclear weapons put together. It killed 75% of all species. And we're used to thinking of the dinosaurs as the great victims because they were big and they took a long time to grow and they had very specialized diets. They couldn't hide very easily. And we're used to thinking of things like mammals and birds as the great survivors because they were smaller, because they grew fast, because they could eat lots of different foods in terms of the mammals, or in terms of the birds, they had beaks, they could eat seeds. Seeds often survive environmental destruction for quite a long time. That's the classic story. But if these lizards survive, that throws a little bit of a curveball here because, yes, these lizards were small. It makes sense that small things could survive, but they were living right in the backyard of the asteroid impact, right near ground zero. And it seems like they grew slowly. They had small numbers of babies. Those are not things you would think of when you think of the survivors of global catastrophe. And in this case, a global catastrophe that was focused very locally on where these lizards lived. So this is a very interesting new study. I think we'll need some time to digest it, but I think it's a great reminder that when the Earth goes to chaos, when there is sudden climate and environmental change, some very strange and unpredictable things can happen.
What a lizard. I mean, this rock fell on its head with immense power.
6-mile
wide
rock. Yeah.
That's a miracle.
No, it's not.
Oh, okay. That's all for this week. You've been listening to The World, the Universe and Us.
Do go ahead and subscribe and spread the word about the show. Give us a rating.
Thanks to all our guests and thanks to you for listening.
We'll see you next week. Bye.
Bye.
Bye.